Hello, everyone. On behalf of Precision Nanosystems and specifically the entire East Regional team, I want to say welcome and thank you for attending our webinar on the benefits of single-use technology. My name is Jason Coleman, and I'm a field application scientist for the Mid-Atlantic and Eastern Canadian regions. I want to introduce the East team and go over a few logistics for how today's webinar will go. As a team, we're responsible for all support in the Eastern US and Canada. We have Pratik Goswami, who is giving the talk today. He is a field application scientist for the New England and Boston region. Once Pratik completes his talk, we will answer your questions and have a discussion. At that point, we will be joined by the remaining members of the East team, including Territory Manager Todd Quitchoff and the Regional Manager for New England area, Shri Kakumanu. In a time where we can all appreciate the importance of minimizing risk and the speed of therapeutic development, I'm pleased to present to you Pratik to talk about the benefits of single-use technology.
Hi, everybody. Welcome to this webinar. So as Jason mentioned, my name is Pratik, and I'll be talking about the benefits of single-use technology, which really focus on the NanoAssemblr platform. So before I go on to that, I wanted to quickly give you an overview of our company. So our company, Precision Nanosystem, started in 2010, so it's been about 10 years for us. And really, we are focused in giving innovative solutions to our customers who are working in different kinds of disease areas, whether it's oncology, rare disease, or infectious disease. Now, talking about our solutions, it can be divided into two parts. So we have our manufacturing technology, which involves the NanoAssemblr platform, and that has the different instruments that we have, and I'll go more in details pretty soon about that. And ancillary to that, we have our nanoparticle technology. So all the different reagents that we offer, along with services like formulation services and clinical service. Now, all of this is really possible because we are a science-based company, so we have really great expertise. We have experts working for us who are focused in nanomedicine development, whether it's microfluidics, engineering, as well as different types of lipids. So what we can do is we can really leverage the breadth of knowledge we have in terms of formulation science and help our customers move along the drug development pathway. So in the drug development pathway, we have the discovery stage, the preclinical, and the manufacturing stage, and we can really come in with our solution at any point here and help you accelerate the research process as you move towards your end goal. Now, when we think about this whole platform, drug delivery pathway and put it against our own NanoAssemblr platform, this is how it would look like. So we have solutions at each step, as I said. So say, for example, you're working on small scale. So you're working on trying to screen different kinds of lipids or maybe different kinds of nucleic acids or cell line. And at that point, you can use our SPART, where you can use 50 to 250 microliters of formulations for screening purposes. So if you have a little bit of a bigger requirement, so say, for example, 1 to 15 ml formulations, that's where the Ignite comes in. It's really our workhorse. So you can use the Ignite for optimizing your formulations. You can change different parameters within the instrument itself, including the flow rate or the flow rate ratio. And the formulation you get out of it, you can either do, for example, in vitro studies for cell lines or in vivo studies in mouse models. Now, if you have a larger animal requirement, say, for example, you're trying to look at non-human primate model or canine model, that's when you have a requirement for a bigger formulation quantity. That's when we move on to the Blaze. So the Blaze, you can do formulations from 10 ml all the way to 10 liters, giving you really a great flexibility there. Finally, when you're ready to move to the clinic, that's where we have our GMP solution. So we work with you at that point, ensuring that we build the system custom made for the particular formulation you're making, so that you can quickly go and have the product out in the market. So again, like I said, supporting all of this, we have our other solutions, for example, the reagents. So an example is the GenVoy. So the GenVoy helps you make lipid nanoparticles, both in the Ignite as well as in the Blaze scale. And then the other solutions we offer is like the formulation solution team. So that team can help you make formulations on your behalf, or actually optimize your formulation on the NanoAssemblr platform. Then we have the clinical team. They can come in at any point, guide you as you work towards the final goal of going to the clinic, and help you doing that transfer when the time comes. Now, one key thing to note here is that for this NanoAssemblr platform, with all of these different instruments, when you're going from one to the other, even though you're changing the scale, it's going to be a seamless transition for you. And the reason behind this is because of the physics or the actual science behind all of this remains consistent. So you have the same microfluidic mixer, which is consistent in all of these different instruments. And the microfluidic mixer that we have is what we call the NxGen Technology. So we have that small cartoon at the left side, and that's what is our NxGen Technology. So what it does is that it enables you to have really precise control of the process of making the nanoparticle, ensuring that you get reproducible result each time. And as I said, because it's true for all the different instruments, it's really scalable, going from really preclinical, pre-discovery stage all the way to the clinical stage. Now, I wanted to go into a little bit more details on this, how this NxGen came about. So I have this slide over here. So this just walks you through the different generations, I suppose, of making a nanoparticle. So we start with the high energy techniques. So that was great as a first technique because, so an example of that would be something like an extrusion method. And because of the way that the whole process is, because it involves higher energy, it does have limited application. And especially, it is really hard when it comes to scaling up to different sizes. The next in line would be an inline macro mixing. So an example would be T-tube mixing. Now this really greatly improves upon the previous generation, so you have much better scalability, but it's still difficult in terms of reproducing the same particles each time. And that brings me to the next generation, which is the microfluidic approach. So the cartoon on the top, that is what we call the staggered herringbone mixer. That is actually the first mixer which we started off with, which was in our initial instrument, which is the Benchtop. So what happens in this microfluidic approach is that the liquids come in a time invariant, non-turbulent manner, and the small crevices in there help in the liquid fold onto itself, thereby greatly accelerating the mixing process. Now what it leads to is that it's really reproducible. So each batch gives you the exact same particles every time. Now it does have some limitations. One key thing is that this architecture is really optimized for about 12 milliliters per minute in terms of flow rate. That is great for normal R&D, but when you're trying to scale it up to larger scale production, that's when that can be a bottleneck. And so the other issue is also with the fact that because of the small crevices in there, it adds complexity in terms of its manufacturing. So then we finally move on to the NxGen. So this is what we developed in-house, and that really builds on to the pre-existing microfluidic staggered herringbone. So it has all the benefits that was there, but it really builds on with the fact that now you can go at a much higher or much faster flow rate. So this can go up to 250 milliliters per minute. So just diving even more deeper into how microfluidics work, I have this particular slide here. And so at the bottom, right-hand side, you'll see the comparison between microfluidics and doing a macrofluidic mixing, like a T-tube mixing. So in a microfluidic mixing, because it is a uniform mixing and the mixing happens really fast, like I said, it happens actually within 1 millisecond. So what happens is you have a homogeneous mixture. Now, when the nanoparticles form, it is all the components coming together in a non-covalent interaction forming those nanoparticles. Now, because it's a uniform mixture, all those local points are the same, and as such, you have the same nanoparticle forming. So you have a great degree of reproducibility. Unfortunately, that's not true for like a macro mixing, because it's a turbulent mixing. All the local points will have different mixing, and as such, you have highly dispersed in terms of the type of nanoparticles you form. The other thing I'll point out is in terms of the limit size. So what is limit size? Limit size means for all the different components you have for forming a nanoparticle, if you take that together thermodynamically, what's the smallest nanoparticle size you can make? So when you're using microfluidics, as you can see on the plot here, which is on the right-hand side, it shows you that you can reach the limit size very early using PNI microfluidics, while if you're doing a macrofluidic, you may never reach that limit size. Now, these are things to really consider when you're building on and trying to incorporate that into your workflow of developing a drug, because when you have it in a PNI channel, once you reach the limit size, even though you increase the total flow rate, you're still at that same point. So you can keep increasing the flow rate. Now, as I said, that becomes crucial when you have larger scale production, especially when you think about, for example, clinical batches. Now, in terms of clinical batches, your end goal is essentially making this production under GMP conditions. The reason you have everything under GMP condition is that you want that every stage should be reproducible. All the things should be reproducible. It should be safe, and you are essentially ensuring that there is no transfer of any kind of contamination from one run to the other. Now, one of the ways GMP has adopted doing this is using single-use technology to get it done. Now, single-use technology ensures that you have the same efficiency and versatility, but at the same time mitigating any kind of cleaning requirements you may have, any kind of cross-contamination, thereby increasing your speed to the market. Now, we really wanted to incorporate all of these highlights into our system, not just into the clinical phase, but also into our preclinical instruments, more specifically the Ignite. So the Ignite essentially brings all these features at volumes which are really low. So you can do formulations as low as 1 milliliters. Now, the Ignite, of course, is built upon the pre-existing instrument that we had previously, which is the Benchtop. So it's kind of like the sequel. So the Benchtop was our first instrument that we introduced in 2014, and it was really successful for the 5 years it was there. So during this time, our engineers and our scientists could take the feedback we got from the users and really build on the system. What we wanted to do is that we wanted to incorporate all the highlights, plus ensure that we have added advantages to using a new system. So that's where we go to the Ignite. So the Ignite has some really critical advantages. The major one is the fact that now it has the NxGen technology. So it gives you seamless transition with all the rest of the lineup. It is also backward compatible with the previous generation mixer, the staggered herringbone that was true for the Benchtop. Also, it is really designed with the future in mind. So it has this additional pump so that right now you can do dilution, inline dilution for your formulation. But down the line, we'd like to introduce more cartridges which can leverage that. Say, for example, doing a double emulsion within the same cartridge would be one thing. Now, apart from all of these great advantages, one key thing which distinguishes the Ignite from the Benchtop is the fact that Ignite now incorporates single-use technology over the multi-use model, which was true for the Benchtop. Now, you may be asking, why do we need single-use technology at this scale, at the preclinical scale? And when we sat down and looked at it, we found that there are actually significant advantages to incorporating this in this particular scale. And these are listed over here. So you can minimize your risk, you really accelerate your program, you can increase your flexibility in terms of where you allocate your resources, and then it really helps in reducing your carbon footprint. Now let's go over each one of them one by one to really impress upon you their advantages. So when you think about single-use technology, minimizing risk might be the most obvious in terms of the benefits, right? Just because since it's single use, there is absolutely no way you can have cross-contamination with the previous run you did. Now that is one issue when you would be using like a multiple use cartridge, just for the fact that you have to really have this additional steps to mitigate that. So you'll have to incorporate cleaning protocols and ensure all of these are adhered properly so that you prevent any sort of contamination. Now, the Ignite cartridges themselves are manufactured in a highly automated clean room. So there is really no way you could have pre-existing contamination, and as such, you are guaranteeing that your formulation is the same every batch each time you do it. Now, when you're doing a formulation in a multiple use format, because you have these additional steps in there, what would happen is that you will introduce, you may introduce human error at each step, right? So because there are additional steps, there is an opportunity that you have contamination in each of these build-up steps. Now, thinking about these steps, going it a little bit more further into that, if you look at the workflow of it, so let's again compare the Benchtop with the Ignite. Now, because, like I said, it has multiple steps in there, so it has the process, so you start with a prime. Now, the reason you want to prime is first to wet essentially the inside of plastics of the cartridge. And besides that, you want to dilute and flush out any kind of contaminant that may have crossed over from your previous run. Then you do your formulation, and after the formulation, you have things like you do cleaning protocols. So normally we recommend that you clean first with the solvent that you did the formulation with, and then you clean it with water. And then finally, you do a purge where you just run air through it. Now, because this is multiple use, at some point, say for example, after every 2 runs, we do suggest that you do some kind of inspection, like a visual inspection, to ensure that the cartridge still has its integrity. And all of these steps are additional steps, right, to the formulation. Now, come to the Ignite, there is only one step. So there is just the formulation step, and that's it. So as you can imagine, you have considerably optimized the workflow, you have reduced it. So we had our scientists actually, who are experts in both this instrument, run this workflow. And on an average, we found that there was a significant difference in time required for formulation using the Ignite versus the Benchtop. So if you look at it in terms of percentage-wise, it's about 70% of boost in terms of productivity. So that amount of time you could spend, say for example, in your downstream processing or analyzing your data. Basically any way that can boost your productivity rather than waiting to get your formulation being done. Now, moving forward, in terms of looking at resources, I just wanted to focus upon, again, taking the example of both the Benchtop and the Ignite. So if you look at, say, for example, per formulation, what is the cost? So the Benchtop, we recommend 5 times uses. So if we do the calculation, you'd find out that actually for per formulation, the price is pretty much the same. Now, if that is same, you now have to think about where else am I spending the resources? So for the Ignite, there is nothing else. But for the Benchtop, because of those additional steps you have, you are spending labor, you're spending time, and then you are also spending resources in terms of solvents you'd use for the different steps like the priming and the cleaning, as well as the additional syringes. So all of these resources you could have definitely allocated for increasing your productivity and reaching your end goal much faster. The final point that I have is the fact that single-use technology can cut down waste. Now, this is one point which, when you first think about it, it can be counterintuitive, like how can single-use be cutting down waste? But you actually, once you do the calculation, it makes sense. So the single-use cartridge, the Ignite right now, it's actually two pieces molded together. It uses much less plastic than the previous generation of the Benchtop, which is 14 pieces combined together. Now, besides that point, the fact of the matter is that this Ignite cartridge, the amount of cartridge plastic is the same as a single 10 ml syringes. Now, I'll take you back to the workflow again. So if you think about the workflow, because the Ignite has just one step, you have 2 syringes and 1 cartridge, which is equivalent to a syringe, so about 3 syringes. But when you look at the workflow for a multiple step, you have the priming, you have the purges, you have the cleaning. All of these steps involve additional 2 syringes, right? So for each formulation, you have about 8 to 10 additional syringes. When you take all of this together, it's about 70% more plastic than you would use when you have a single-use technology. So that really means that you can reduce your carbon footprint when you go via the single-use technology route. So tying all of this together, just in summary, I wanted to go over each again. Saying that single-use technology can really help minimize your risk. You are mitigating the risk at the one step, so it's just a single step, so no risk of cross-contamination at all. Then you can really accelerate your program. Again, single step, you do not have to do these additional steps. You can increase your flexibility in terms of allocating your resources better because you can cut down on both the labor and the time. And finally, with the less amount of plastic that you are using, you can reduce your carbon footprint by using single-use technology. Now, using single-use technology in conjunction with the other improvements like the NxGen technology can really help you accelerate your timeline in whatever scope you're working in. So for example, if you're in the pre-discovery stage, you can really accelerate by using, again, the Spark and the Ignite. Or when you are in a more advanced stage, you can accelerate even further ahead when you go to the Blaze or to the GMP system. Well, with that, I'd like to thank everybody for listening in, and I turn it over now to Jason.
So I just want to say thank you to everyone. But yeah, so I just want to close out again and say thank you to everyone. So thank you.
Thanks, everybody.
Everybody.
Thanks a lot.